Welcome to the Clinical Omics webinar series. Our presentation today is entitled Genetics of Neurodegenerative Disorders, How Mutations Affect Disease Risk and Pathology. I am Chris Anderson, Editor-in-Chief of Clinical Omics, and I'll be the moderator for today's webinar. Mutations in specific genes have been shown to alter disease risk, progression, and pathology in various neurodegenerative disorders. Today's presenters will look at two of the more common and investigated neurodegenerative afflictions, Alzheimer's disease and Parkinson's disease. While there's been significant work to define specific genes linked to the risk of developing these diseases, today researchers are increasingly focusing on the symptoms of disease progression and the role played in this by the presence of Lewy bodies and specific genetic alterations. Let's meet our speakers today, who will detail their findings on specific genetic markers, APOE E4 for Alzheimer's disease and GBA for Parkinson's disease. Byron Creese is a senior research fellow at the University of Exeter in the UK. Dr. Creese's current focus is a project using genomic data to identify genes and molecular pathways associated with key neuropsychiatric symptoms in dementia, with a view to identifying potential disease mechanisms underlying these symptoms. Dana Sullivan is the Marketing Product Manager at Canon Biomedical. Sullivan's work at Canon focuses on the company's novel genotyping assay. The last presenter today will be Dr. David Munoz, a professor in the Department of Laboratory Medicine and Pathology at the University of Toronto. Dr. Munoz's research is focused on neuropathology, particularly the mechanisms of development and progression of degenerative diseases of the brain, including Alzheimer's disease, the non-Alzheimer dementias, movement disorders, and amyotrophic lateral sclerosis. Before we get started with the presentation, I want to encourage everyone to submit questions for our Q&A session at the end of the presentation. We will try to answer as many questions as we can, so simply type your question into the Q&A box on the left-hand side of the screen and hit submit. Okay, with all the housekeeping taken care of, let's get to the presentations. Byron Creese, take it away.
Hello, good morning. I'm going to talk to you today about the relationship between GBA mutations and Parkinson's disease, with a particular focus not just on risk for Parkinson's disease, but also on the progression of Parkinson's disease with respect to neuropsychiatric symptoms. So most of what I'm going to talk to you today about relates to this publication, which came out in Neuropsychiatric Genetics earlier this year. And there's the link for those of you that are interested. Okay, so the GBA gene. So this is the gene that encodes for the lysosomal enzyme GCase. Homozygous mutations in this gene cause the lysosomal storage disorder Gaucher disease, GD. There are three types of GD. Type 1, which is non-neuropathic GD, and types 2 and 3, which are neuropathic GD. Different mutations lead to those different types of GD, and I'll talk a little bit more about that in just a moment. But firstly, why are we interested then in the role of these mutations in Parkinson's disease? Well, there is an increased rate of prevalence of Parkinsonism in GD, and indeed also in the relatives of individuals with GD, and this led to a focus on a putative role for these mutations in Parkinson's disease. And indeed, we now know that heterozygous mutations in GBA are one of the biggest genetic risk factors for both Parkinson's disease and dementia with Lewy bodies, and those two neurodegenerative diseases share the same hallmark alpha-synuclein pathology. So GBA, or the GBA gene, is a highly polymorphic gene. There are over 300 mutations in this gene, and I've just listed a few of them on this diagram here. In red there, you'll see two of the most common. That's L444P and N370S. You may see them, I should say, you may see them referred to as neuropathic or non-neuropathic, and that refers to the type of GD that they are associated with. So L444P results in neuropathic GD. You may see this referred to as a severe mutation as well. N370S leads to non-neuropathic type 1 GD, and you may see this referred to as a mild mutation. There are also mutations which do not cause GD, but there is evidence that they increase risk for PD. And those are just the two that are highlighted, or two of those are highlighted in blue right there. So the point there is that there are mutations where the clinical consequences are not entirely clear, but we do, when we're thinking about GBA in Parkinson's disease, we do need to probably think beyond those mutations that cause GD. So L444P and N370S, which I highlighted on the last slide, are among the most common GBA mutations, although they are still themselves pretty rare in most ethnic groups. They are, as shown in this table, they are most common among Ashkenazi Jewish people of Ashkenazi Jewish ancestry. We can then see among East Asian populations, this particular Japanese study reported a fairly high frequency of a variant that's not really seen elsewhere, but otherwise in East Asia pretty low. Same goes for European populations, but individuals of sort of Iberian, Portuguese, Spanish ancestry seem to have a slightly higher frequency as well. But overall, these mutations are quite rare, which makes studying their effect a little bit difficult, which is why meta-analysis, which I'm going to talk about later, is a good technique to use. This review here is a pretty good review of all things GBA, so I'd recommend reading that for anyone who wants some more information. We know that GBA mutations are associated with Parkinson's disease from the previous slide. So with that in mind, we had a look at some of our samples in our biobank. We had 112 Lewy body dementia cases, 76 Parkinson's disease cases with psychosis, and 400 Parkinson's disease cases unselected for psychosis, so some would have them and some without. Here are the frequencies for the GBA and 370S mutation in each of these groups. We have around about 4% frequency in the Lewy body dementia cases, higher than that, 8% in the PD psychosis cases, and around 1% in the Parkinson's disease cases. So these are all Caucasian European samples. And what we see here is that there is a higher frequency among the dementia cases, which include Parkinson's disease dementia and dementia of Lewy bodies. And again, the Parkinson's psychosis cases have a higher frequency than Parkinson's, than an unselected Parkinson's population. So this is just preliminary data. It's nothing conclusive, but it was enough to get us thinking, in addition to the existing literature, get us thinking along the lines of characterizing the role of GBA in the progression of Parkinson's, particularly with respect to neuropsychiatric symptoms. So I've just explained in the last couple of slides that Parkinson's disease, or GBA, sorry, is a risk factor for Parkinson's disease, and there is a growing body of literature, particularly around dementia and cognitive impairment, that GBA mutations also influence disease progression, and there's a bit of preliminary evidence from our own cases, but also there's more robust evidence in the literature of a relationship between psychosis as well, the development of psychosis as well. So the question then is, what is the mechanism? Well, the first thing to point out is that GBA is not a risk factor for Alzheimer's disease, so it's unlikely that the mechanism by which GBA is increasing risk for these symptoms is via AD processes. We know that, or certainly it appears that there's a relationship between GBA and alpha-synuclein aggregation, and there are gain of function, loss of function hypotheses to explain that. No unequivocal evidence, as far as I'm aware, to lead to the rejection of either of those, but there's quite a bit of literature in the basic science base to support both positions. There are mouse models of Gaucher disease which carry homozygous mutations in GBA, and they've been shown to exhibit memory deficits and hippocampal alpha-synuclein pathology, which of course is important if we are looking for mechanisms of cognitive impairment and dementia. The same level of cognitive deficit is not present in heterozygous animals, but extensive cognitive phenotype characterization I think actually is probably underway by some colleagues of mine at King's College in London. And this, of course, will be an important avenue of future research, because heterozygous animals are going to be a better model for PD and DLB, because it's heterozygous mutations that elevate the risk in those diseases. Beyond animal models, we have seen in recent years the first use of stem cell technology to understand how GBA mutations in dopaminergic neurons influence alpha-synuclein levels, and we've seen in one study they're associated with higher extracellular alpha-synuclein. Other studies have shown GBA mutations being associated with a series of disruptive lysosomal processes, and in vivo, in humans, GBA activity in blood has been shown to be reduced, and plasma oligomeric alpha-synuclein elevated among GBA mutation carriers compared with non-carriers. So it seems like GBA mutations are associated with fundamental Lewy body disease pathology. We know that they are a risk factor for Parkinson's disease and dementia with Lewy bodies itself. And we also know that GBA mutations have been shown to negatively impact motor and autonomic symptoms in PD as well. But here we want to focus on the emerging evidence around psychiatric phenotypes. So neuropsychiatric symptoms in Parkinson's disease are very common. They're associated with worse disease outcomes, and they are a major cause of distress for patients. Their underlying biology is only partially understood, so it is important that we attempt to find factors which will allow us to both understand a little bit about which groups of patients will develop these symptoms, and also about a little bit about how we understand the mechanisms with a view to improving treatments. Now, genetics is one way that we can understand those mechanisms, and looking at GBA, we hope, will help shed some light on those. So we started off thinking about this by conducting a couple of descriptive reviews. These came out earlier this year in Nature Reviews Neurology. This is a relationship between, or just a table summarizing the relationship between different polymorphisms in different genes and cognitive impairment or dementia in Parkinson's disease. And I won't go into it in detail, but suffice to say the most consistent evidence across all these different genes was for GBA mutations. In the same journal, we also published a review of genetics of psychosis in Parkinson's disease, and again, we found the most consistent evidence for GBA mutations. Now, this is interesting because we do not often see such strong associations with single genes for complex traits like psychosis. And I think this is an important point, and it relates back to what I was saying about the fundamental disease pathology associated with GBA mutations. I think it's important at this point not to consider these symptoms in a siloed way, but think about that psychosis and cognitive impairment are part of the same more severe disease course that is associated with GBA mutations. And I'll return to that a little bit later. So we wanted to, instead of, or off the back of these descriptive reviews, wanted to think about quantifying the risk attributable to GBA mutations with respect to the development of key neuropsychiatric phenotypes in PD. Those neuropsychiatric phenotypes were chosen to be cognitive impairment, psychosis, and depression, because those are three of the most common that are looked at, and we could be sure to identify a sufficient number of studies to conduct a meta-analysis of risk. Other inclusion criteria were fairly broad: diagnosis of idiopathic PD, written in English, and we excluded studies that were restricted to familial Parkinson's disease only. So the first symptom that we looked at was cognitive impairment. Specific inclusion criteria here were that studies did not exclude people for displaying cognitive impairment. They used an assessment of cognitive impairment either by operationalized criteria for MCI or dementia, or by falling below a specified cutoff on a standardized assessment tool. That's to ensure a robust definition of cognitive impairment. Inclusion criteria were cognitive impairment not being treated as a dichotomous outcome variable. So that's to say we're interested in, is there cognitive impairment or dementia, yes or no, rather than a continuous scale. And any studies which didn't report the frequency of cognitive impairment would mean that we can't do the meta-analysis. So we were not able to include those. In total, there were 13 studies, I think, that met our inclusion criteria. And among those studies, the blue shape at the bottom there is the one to pay attention to. That indicates that across all these studies, we found a 2.5-fold higher risk of cognitive impairment or dementia in PD associated with GBA mutations. So this is among around 400, just over 400 GBA characters and around carriers, and around 6,000 non-carriers. A more detailed review of these studies highlights some interesting detail. So one study found that there was looking at more detailed neuropsychology, there's possibly a greater burden of executive functioning deficits relative to language deficits among GBA carriers. And another study found some evidence of differential effects of mutations. So if you recall what I mentioned at the beginning, L444P being a severe mutation, N370S being a mild mutation, one study found that indeed severe mutation carriers were more at risk of dementia than mild mutation carriers. And also in neuroimaging studies, there were different patterns of cortical activation associated with severe mutation carriers, those patients actually having a more similar phenotype to DLB, dementia with Lewy bodies, rather than Parkinson's disease. The difficulty with doing these studies is you need a very large sample size, but it probably is quite an important avenue for future research. Moving on to psychosis, the inclusion and exclusion criteria broadly similar to cognitive impairment and dementia, in the sense that we're looking at a reliable and solid evaluation of psychosis, meaning hallucinations or delusions. We want either a yes or no classification, and the exclusion criteria reflects that as well. We found 6 studies here meeting those criteria, and among those we find a nearly 2-fold higher risk of psychosis in Parkinson's disease. So that's the first time that, to my knowledge, that this has actually been quantified. Importantly, it's difficult to know what the relationship here is with cognitive impairment. So we know in Parkinson's disease that psychosis and cognitive impairment are closely related. They're both, cognitive impairment is present by definition in dementia, but psychosis and more severe psychosis is strongly associated with more severe cognitive impairment, and psychosis as well associated with more rapid cognitive decline. So I think it's important just to come back to that point, that I'm not necessarily talking about specific risk for psychosis and unique mechanisms giving rise to psychosis. It could be the case that GBA is driving fundamental disease pathology, which gives rise to this more severe Parkinson's phenotype. So the last symptom we looked at was depression, and again, I won't go through these inclusion criteria because they are basically following the same pattern as the last 2 symptoms that I spoke about. So again, what we're finding among these six studies that looks at depression is a round about 2-fold higher risk of depression in PD associated with GBA mutations. We would advise interpreting this with a little bit of caution. The meta-analysis diagnostics were showing some evidence of publication bias. So that's to say that across these 6 studies which met our criteria, they're all in a positive direction, and normally we wouldn't really expect that. We'd expect a little bit more variability, even if the overall result is a positive relationship. Reflecting that wider review of these studies, of more studies, not included in the meta-analysis, does paint a more nuanced picture. Specifically, there were a number of studies which did not find an association with depression. We didn't include them in the meta-analysis because they didn't treat depression in the way that we required. So depression was measured on a continuous scale rather than a binary outcome measure. But it's important to point that out. I think this will be because depression, or at least in part because depression is a much more complex genotype. It's probably much more multifactorial in its etiology. So we may have depression resulting directly from neurodegeneration, but also if GBA mutations are associated with a higher burden of motor and autonomic symptoms, depression could arise as a sort of psychological consequence of having to handle that element of the disease. So to summarize, neuropsychiatric symptoms are very important in Parkinson's. We need to focus on them. They're common, distressing, challenging to treat. And we have now shown that there are a complex and broad range of symptoms associated with GBA mutations in Parkinson's disease, not just the motor and autonomic, but extending into neuropsychiatry, as we have shown. Now, mechanistically, there's evidence from a various array of basic science studies pointing to GBA mutations driving alpha-synuclein aggregation, although the exact mechanisms remain to be established. There are studies showing a greater evidence of cortical and hippocampal Lewy body pathology post-mortem, although that's not been sort of conclusively proved. But that idea is consistent with the expression of these neuropsychiatric phenotypes. However, I just want to end again with this point that it's also indicative really of a broader clinical GBA PD phenotype, which is characterized by more severe disease course across a number of domains, which now include neuropsychiatric phenotypes as well. Thank you for listening.
Thank you, Byron. A very interesting conversation on the broad range of symptoms associated with GBA mutations in Parkinson's disease. A quick reminder before we move on to the next presenter that we will be having a Q&A session at the end of today's webinar. All you need to do is type your questions into the Q&A box on the left-hand side of your screen and submit the question. And now on to Dana Sullivan of Canon Biomedical.
Thank you for joining our webinar today. I would like to thank Dr. Munoz and Dr. Kreis for presenting on newly associated research for the GBA and APOE genes. My name is Dana Sullivan, and I am Product Manager at Canon Biomedical. If any of you listening are interested in investigating mutations within either the APOE or GBA genes, or need a simple method to genotype a particular neurology-related target, Canon Biomedical has developed a genotyping library of research-use-only products linked to many diverse fields of human biology, including neuroscience.
These assays with master mix use fast and simple PCR followed by high-resolution MELT analysis, HRM, and provide an easy method to detect mutations of consequence.
HRM analysis distinguishes DNA variations by analyzing sequence-specific MELT curve patterns. It can be completed in under an hour and is a targeted, rapid solution as compared to sequencing, which can be costly and takes much longer. Data analysis requires a simple visualization of MELT profiles and simplifies experimental design by not requiring a minimum number of samples to distinguish the genotype. That means with HRM, one sample can be run with known controls to accurately genotype a single sample. Canon Biomedical offers over 80 SNP and indel assays specific to neuroscience and associated neurological diseases. On this slide, you can see a handful of the targets we offer and the specific diseases they associate with, including mutations in GBA, those discussed by Dr. Kreis today, and in APOE, soon to be presented on by Dr. Munoz. The two APOE assays that Canon Biomedical offers helps distinguish APOE alleles. Together, they detect single nucleotide polymorphisms in APOE gene that causes an amino acid change based on the variation of mutation. Dr. Munoz will be discussing APOE in more detail in the next presentation. But before that, I'd like to go over a few things. There are three common APOE alleles, APOE 2, 3, and 4. E2 is relatively rare and may provide some protection against the disease. APOE 3 is the most common allele. It is believed to play a neutral role in the disease, so neither decreasing nor increasing risk, and considered the wild type. E4, which will be discussed in even more detail by Dr. Munoz, increases risk for Alzheimer's disease. For a little background, everyone has two copies of the gene, and the combination determines your APOE genotype. The APOE2 allele is the rarest form of APOE, with an allele frequency of approximately 8% in the general population. And research shows that carrying even one copy appears to reduce the risk of developing Alzheimer's by up to 40%. Research has also shown that the APOE4 allele is present in approximately 10 to 15% of the general population, but present in 40% of people with Alzheimer's disease. Having the allele increases the risk for Alzheimer's and lowers the age of onset. Having one copy of APOE4 So for example, if you have the genotype E3E4, can increase your risk by 2 to 3 times, while having 2 copies, E4E4, can increase the risk by 12 times. The 2 novel APOE assays developed by Canon Biomedical can be used to determine a sample's APOE genotype. On this slide, you can see data from blood samples tested with one of the APOE assays I'm speaking about. You can see that all 3 genotypes, wild type, homozygous mutant, and heterozygous, are easily distinguishable based on their MELT curves. We also offer characterized genotyping controls for genotyping assays, including our APOE assays. A novel control set contains a homozygous wild type and a homozygous mutant control. A heterozygous control is formulated by combining the 2 provided controls. On the right-hand side of the slide, you can see that by using our novel controls, you can compare sample curve data to control curve data to call the sample's genotype.
These 2 APOE assays and other genotyping assays, master mix, and controls are available direct in the U.S. and via distributors throughout Europe and Canada. If you are U.S.-based and are interested in purchasing or discussing any of the products offered by Canon Biomedical, please email contact@canon-biomedical.com. You can also learn more about the targets and other products we offer by visiting our website, www.canon-biomedical.com. Before I close, a few disclaimers. All products I have discussed are research use only. For licensing information and user guides, you can visit our website. And lastly, Novolil is a trademark of Canon Biomedical.
Once again, thank you for attending our webinar today. I am happy to answer any questions at the close of the webinar and look forward to hearing Dr. Munoz present more on APOE.
Thank you, Dana, for that brief presentation on genotyping solutions for APOE in GBA. Again, a quick reminder that there's a Q&A at the end of our webinar today. Simply type your question into the Q&A box on the left-hand side of your screen and hit submit. And now our final presenter, Dr. David Munoz. David, take it away.
Good morning. We will be talking today about one aspect of APOE in Alzheimer's disease, which is not directly related to its well-known role as a risk for the disease, but its effect on psychosis in Alzheimer's disease. APOE comes in 3 different alleles, E2, E3, and E4. E3 is the most common and considered neutral. Of course, each person receives one allele from the mother and one allele from the father, so we have different combinations. The graph on the right represents the probability of developing dementia from people as they age. The vertical arrow represents the probability for someone with an APOE genotype 3-3. The horizontal arrow represents the probability of developing dementia for someone with a genotype 4-4. As you can see, with that genotype, almost everyone will be demented by age 78. This is a different way of expressing the risk related to the E4. What is the difference between the different alleles in their actions relating to Alzheimer's disease? One thing we know for sure is that E4 is far less effective than E3 or E2 in the clearance of beta amyloid. This is probably not the only difference, because we know that there are structural and functional changes in the brain in patients who will develop Alzheimer's disease well before there is any detectable accumulation of A-beta. In this slide, we have in subjects who are cognitively normal, so before developing any disease, different groups, some of whom the three groups on the left do not have any amyloid, whereas the groups on the right have amyloid. The second panel shows the frequency of E4 allele, which, as you can see, is much greater in the groups that have amyloid. Interestingly, in the middle, you see a tiny green, which represents subjects who have neurodegeneration and tau deposition, but no amyloid. This is clearly a group that is not related to APOE4. An important recent development is that APOE influences not only the risk of developing Alzheimer's disease, but also the probability of having Lewy bodies in patients with Alzheimer's disease. We now know that sex is a major modifier of the effect of APOE in relation to the risk of developing Alzheimer's disease. The graph on the left represents in red females, the solid line, those who are non-carriers of E4 versus the dotted line who are the carriers. The blue lines represents the males. As you can see, the difference between the carrier versus non-carrier females is greater than the difference between carriers and non-carriers males. On the right, the graph represents E3 in the solid line versus E3 E4 on the dotted line. Interestingly, while in females the difference is highly significant of the effect of APOE on the risk of developing AD, in males there is no significance. It does not reach the threshold for significance. Delusions are a false belief in which the patient is not only suspicious but actually convinced that this thing has actually happened. And you can see the kind of questions that you can ask the patient to indicate whether he or she is having delusions. Okay? For example, if someone is stealing from them, or someone is living in their house, or that their house where they're living is not their home. We have used a data mining approach utilizing the National Alzheimer Coordinating Center, or NACC, because of the abundance of cases that is what we need to be able to make the approach that we use. The NACC has a database, and it has information that is clinical, cognitive, behavioral, and many of these patients are followed to autopsy, and the autopsy is performed in and reported in standardized manner. The Neuropsychiatric Inventory, or NPI, is administered by the clinician to the caregiver, not to the patient, and is scripted. That is, the questions must be asked exactly as listed, and if the answer to any question is yes, then the responder is asked to rate the frequency and severity of the symptom. It has been established as a reliable test that has concurrent validity and, most important, convergent validity. That is, that the items in the test relate to, for example, regional atrophy or hypometabolism, PET, as well as autopsy studies of plaques, tangles, and neurochemical parameters. The NPI has 12 items, of which the first 2 are hallucinations and delusions, which are the ones that we will focus on today. But their questions also involve other issues like agitation, apathy, aberrant motor behavior, or sleep disturbances. The NACC utilizes the NPI-Q. Q is for quick, so that's a short version. And the questions specifically asked are for delusions. Does the patient has false beliefs, such as thinking that others are stealing from him, her, or planning to harm him or her in some way? And for hallucinations, does the patient have hallucinations such as false visions or voices? Does he or she seems to hear or see things that are not present? To evaluate the pathology, we considered senile plaques, marked here with a blue arrow, and neurofibrillary tangles, marked with a red arrow. And we use 3 semi-quantitative measures of Alzheimer pathology: the Braak stage for neurofibrillary tangles, the CIRAD neuritic plaque density, and their combination, which is known as the NIA rating. In addition, we looked at other pathology, including Lewy bodies and vascular pathology. The Braak and Braak staging is based on the extension of neurofibrillary pathology and runs from the stages 1 and 2, which the patient is usually asymptomatic, and the neurofibrillary pathology is limited to the entorhinal areas, and you see that on the left row. Stages 3 and 4, limbic, in which the disease extends in the basal temporal lobe and adjacent regions, and you see that in the middle row, and the isocortical, in which the disease extends to the entire cortex. The CIRAD score relates to neuritic plaques, which are assessed in 3 cortical regions, and age is not considered. The NIA Reagan combines the 2, and when the patient has a high Braak stage and a frequent neuritic plaque CIRAD score, it's said that has a high probability that the dementia is due to AD. This is equivalent to say that has severe Alzheimer pathology load. We wanted to look at the gender and pathology-specific effects of Apolipoprotein E genotype on psychosis in Alzheimer's disease. This is work largely done by my student Julia Kim. For the Lewy bodies, although we can see them on H&E slides as shown on the left panel with the blue arrow, they are easier to detect, and this is the method used with immunostaining for alpha-synuclein as seen on the right panel. As I indicated, I would use the NIA Reagan. Those are only the patients that we use were those with severe Alzheimer pathology. That is both high neuritic plaque density and high BRAC neurofibrillary tangle stage. The exclusion criteria were patients with Down syndrome, Huntington disease, or CNS neoplasm. So we ended up with 1,517 patients with high probability of Alzheimer's disease based on the pathology. That is, all these patients had severe Alzheimer's disease pathology, and their clinical status had been assessed within the 2 years prior to death. We divided this group into patients who had no APOE4, those 622 who had one copy of APOE4, and 201 who had 2 copies of APOE4. Within each group, we divided into those who did not have psychosis and are entered as P-, and those who did have psychosis who are entered as P+. And within those who have psychosis, we divided into those who have delusions, whether they also had hallucinations or not, and those who had hallucinations, so that regardless of whether they also had delusions, so that the group with delusions and the group with hallucinations add to more than the group with psychosis. And we did this for all the groups of APOE4. This graph represents the results as the proportion of subjects with 0, 1, or 2 E4 alleles. On the left, in the group labeled -P without psychosis, and on the right, in the group labeled +P with psychosis. Further right, we divide this group into those with delusions and hallucinations. But the interesting comparison is the one marked by the blue arrows, which show that patients with psychosis have an increased frequency of a double dose of E4, whereas a single dose has little or no effect. If we now stratify by gender, we will find that the difference between non-psychotic and psychotic patients in the frequency of a double dose of E4 is increased in females, whereas there is no difference in males. Dementia with Lewy bodies is a different disease, characterized, among other things, by prominent visual hallucinations. However, Lewy bodies are often seen in Alzheimer's disease, in fact, in as many as 50% of the cases. The Lewy bodies can be recognized using antibodies against alpha-synuclein, as shown on the figure on the left by the blue arrow. It was recently shown that having 2 copies of the E4 allele significantly increase the frequency of Lewy bodies in patients with Alzheimer's disease. In our group, we found that this difference is only present in females. we can now stratify the subjects by presence or absence of Lewy bodies. If we consider just the group with Lewy bodies, the proportion of patients with a double dose of E4 in the psychosis group has markedly increased over the non-psychotic group. This is true both for delusions and hallucinations. In contrast, in the group without Lewy bodies, the presence or absence of APOE4 makes no difference in the development of psychosis. If, in addition, we stratify by gender, females with Lewy bodies who have psychosis are much more likely to have 2 copies of the E4 allele. In contrast, males APOE4 has no effect. And in the absence of Lewy bodies, E4 has no effect on the development of psychosis in either males or females. This is a summary graph. The first row represents all subjects. The second row, those with Lewy bodies. The third row, those without Lewy bodies. The first column represents all subjects. The second column, females. The third column, males. If I may call your attention to the central panel representing females with Lewy bodies, there you can see the marked difference between those with psychosis and those without psychosis in the proportion of the double dose of E4. To summarize these results, we can see that APOE4 is involved in the formation of Lewy body pathology and in psychosis in a gender-specific manner. We know that APOE determines the presence of Alzheimer pathology in a co-dominant manner, and we had previously shown that the severity of Alzheimer pathology does not relate to the presence of psychosis in patients with Alzheimer's disease. In the bottom row, you have what we consider is the mechanism that APOE4 acting in a recessive manner determines the presence of Lewy body pathology, and the presence of Lewy body pathology is associated with psychosis. In a second study, we have looked at the interaction of the E4 genotype and psychosis genotype on clinical outcomes, and this has work done by Winnie Kian. And we compared in a similar group, also distributed in the manner that you have seen before, the effect of APOE first on the mini-mental scores. And as could be expected, patients with the E4 allele, just being curious, having 1 copy is enough, have a lower mini-mental score than patients without the E4 allele. If we look at the effect of the psychosis group with the mini-mental, we see that patients without psychosis have a better mini-mental than patients with delusions, or patients with hallucinations, or patients with delusions and hallucinations, and these differences are significant. If we look at the functional scores, which is measured by the FAQ, and the FAQ is measured in the opposite way, so that the lower, the better. Again, patients without psychosis have a lower FAQ, that is, have better function in normal life than patients with delusions or hallucinations or both. Interesting part is the interaction of APOE with psychosis on these functional measures. So here we're comparing the mini-mental in patients who are either non-carrier or carrier of the E4 allele, and who also are either psychosis-free or have delusions or hallucinations or delusions and hallucinations. The interesting point is that the group that has hallucinations only has a markedly different interaction. That is, in that group with hallucinations only, having one copy of the E4 allele markedly decreases the mini-mental scores. So, in conclusion, the APOE genotype plays roles in Alzheimer's disease beyond the increased risk. It has a recessive, female-specific effect of psychosis mediated through the development of Lewy bodies, and has a dominant effect on the inverse association of hallucinations and mini-mental scores. Thank you.
Before we start the Q&A session, I want to let everyone know this is your final chance to submit your questions for any of our speakers. So hurry up and submit them now. Okay, let's begin the Q&A. We've already received a few questions, so why don't we start in, and we'll try to get to as many as we can. Okay, thank you everybody for joining us for the presentation today. Let's get to our first question. This question is for David. David, what do you mean by codominant versus recessive effect of APOE E4?
Okay, if we consider the effects of APOE on the risk of developing Alzheimer's disease, having a single copy of E4 increases the risk, as we know, approximately 3 times, and having 2 copies increases the risk many more times, approximately 15 times. However, when we look at the effects of APOE E4 on the risk of developing psychosis in Alzheimer's disease, having a single copy has no effect at all. It's only when you have 2 copies that the risk is substantially increased. In fact, in women, particularly those who have Lewy bodies, by 300%. I hope this is clear.
Thank you. The next question is for Byron. Byron, can you elaborate a little more on the relationship between mutations and the emergence of dementia in Parkinson's disease? Hello, Byron.
Hello? Hi.
Hello.
Can you hear me?
Yes.
Yeah, sorry, I did hear the question. In our meta-analysis, we looked at cognition and dementia together, because there was good evidence that whichever way you look at it, there was an increased risk associated with GBA. But with dementia specifically, I'd say there's a couple of important points that I didn't cover. The first one is certainly there does appear to be this relationship between severe mutations having a stronger risk for dementia specifically when compared with mild mutations. Mild mutations also carry an increased risk relative to non-carriers, but severe mutation carriers carry an increased risk both relative to non-carriers and relative to mild mutation carriers. And the second point that I think is probably important is, as well as increasing risk for dementia, there's a good body of evidence showing that actually the time from disease onset to developing dementia within Parkinson's disease is shorter among GBA carriers. That actually goes for dementia as well as onset of psychosis, and indeed mortality as well is also shorter among carriers. Thank you.
Great, thank you. The next question is for David. David, what is the implication of the E4 interaction with hallucinations in determining MMSE scores?
Well, it is not always easy in a demented patient to separate what is a delusion and what is a hallucination. That is, the patient may have a belief that is not based on reality because he or she is seeing or hearing that information. Now, we were surprised to find that the interaction between the APOE genotype and the mini-mental varies depending on the type of psychosis the patient has, so that only in those patients with hallucination, having the E4 genotype is associated with a marked decrease in the MMSE. And that indicates to us that that group of patients, those who have hallucination only, are different. There is a different biology, possibly related to the presence of Lewy bodies. At this point, we do not know if the answer is that.
Okay, thank you. And we do have time for one more question. This last question is for Byron. What use, if any, are GBA mutations likely to have for clinical screening?
Probably not very much. There's a couple of reasons for that. Firstly, these mutations are very, very rare, but also the penetrance is quite low. So that's to say that most people with GBA, heterozygous GBA mutations, don't get Parkinson's disease. So that really limits the use that they can have in terms of predictive value. But that's not to say it's not important that we continue to try and understand what this GBA PD phenotype seems to look like and what the implications are for patients.
Excellent. Well, that will wrap up our presentation today. I want to thank all the attendees for joining us today. Our webinar will be available again on demand, and you will be receiving a link for that. Again, thank you for joining us today, and thank you to our presenters, David Munoz, Byron Crease, and Dana Sullivan, for this fascinating webinar. Have a good day, everyone.
Thank you.